Good morning, everybody.
Good morning.
Howdy, howdy, howdy. Let's bring up the chat. Okie dokie. So, while we get our brains warmed up, anybody got any questions about what we covered last class? So, sex determination, sex linkage. Oh, that looks like a lovely spot, Jesus. Stuff like that. Before we get cracking, time for my caffeine to have effect. Okay.
I have a question.
Fire away.
Can you hear me? Okay, I didn't really get the sisterless and deadpan thing. If you can just go over it real quick one more
Oh,
time.
for sure. So, where were we? Around here, I think. Here we go. So essentially, to be able to determine if you're going to be a male or female cell in Drosophila, and hence male or female fly, you have to figure out how many X chromosomes you have to autosomes. That's the key. That's how they do it. And so essentially what they're doing at a cellular level is comparing quantities of protein. Deadpan, which is produced from the autosomes, and sisterless, which is produced from the X chromosome or autosome. It's only on one autosome. So essentially what the cells are doing is seeing how much of sisterless do I have versus deadpan, or vice versa. Does that make sense?
Yes.
It's like a comparative deal, essentially. And the way they do that is that sisterless stops deadpan from working. So if you have an equal amount of the two, if you have an equal number of X chromosomes to autosomes, those two cancel each other out. So deadpan can't do its job, which is to turn off sex lethal. And so if it's not, essentially, that's really the trick, right? Or not the trick, the mechanism. So if you're female, those two proteins essentially cancel each other out. So there's no deadpan around to turn sex lethal off. And so sex lethal is on, which equals females. Now, if you have an imbalance between those two, right? And the only way you're going to get that imbalance is if you have fewer X chromosomes where sisterless is produced, or from which, compared to autosomes. So if you have only one X chromosome and two autosomes, or that ratio, then there's not enough sisterless. It's like kind of, if I was to do like comparative deal, right? There's not enough sisterless to fully cancel out deadpan. And so deadpan is able to turn off sex lethal. And with sex lethal off, you have males. So it's really a comparison between those two proteins, those gene products. If they're an equal amount, which you can only get if you have an equal number of chromosomes, or sex versus autosomes, they cancel each other out. So sex lethal stays on. If you have an imbalance, and you only get an imbalance if you have fewer X chromosomes than autosomes, then deadpan is still able to do its job, which is to turn off sex lethal. So it's essentially, it's a comparative deal, which then results in an outcome, which is sex lethal on or off. On equals females, off equals males.
Okay, thank you.
Good question. And it's not a simple kind of mechanism to get, but if you keep in mind the whole ratio deal or the comparative aspect of it, it makes a lot more sense. It's a lot easier to grasp. Any other questions? Oh, good stuff. Okay, so what we're going to crack on to here, I don't know why that's there. Here we go. Is essentially other aspects of non-Mendelian inheritance. Right, and so one reason why we spend a bunch of time on Mendelian inheritance, even though it doesn't really apply to really much human genetics other than like disease, congenital diseases, things like that, is that you have to understand Mendelian inheritance to understand non-Mendelian inheritance, right? And non-Mendelian inheritance is the interesting stuff, right? So that's what we really want to kind of dig our teeth into. Right, and so there's all sorts of different types. We're not going to cover all of them, but most of them. So you have, we've already done kind of, well, actually, and in no particular order too. This is just what I remember them in. So you have gene bioenvironment interactions, right, which is the whole nature versus nurture deal, and it's very rarely a all of one or all of another situation. We have differences in dominance, and that also is a matter of scale too, right? So a lot of what we're talking about with non-Mendelian inheritance depends on the scale at which you're looking at it, right? Some things appear very simple when you kind of zoom out and look at it on the larger scale, and it gets much, much more complex as you zoom in towards the cellular and molecular scale. What else is there? Boop-doop-doo. Variable penetrance, so expressivity. So essentially you can have the same genotype as someone else, but display a different phenotype, right? Which is a bit of a head twister. There's epistasis, which is particularly interesting. It's actually something that I use as a tool a lot. Actually, most of my work over the last, I don't know, 9 years or so, I guess, 10 years, can't remember actually, has been using epistasis as a tool to figure out genetic pathways, right? So it's an interesting phenomenon in and of itself, but it's also a very, very handy tool. And actually I got reviews for a little paperback the other day. One of the reviewers says, "Oh, you should do some epistasis experiments." I was like, "Yeah, I know I should, but I haven't on this one anyway. Always too many things to study." And what else? Oh yeah, the major one, polygenic traits, right? And so almost all, again, other than in humans at least, disease-causing traits, right? So, you know, like cystic fibrosis, or sickle cell disease, or what was it, beta thalassemia? You know, there's a whole bunch of them, right? Essentially, if you have a non-functioning gene or 2 copies of non-functioning gene, you have that disease, right? That's pretty much it for humans. Eye color, many genes. I don't know actually how many, 5 or 7. Hair color, 3 to 4 genes at least, right, that I know of. Skin color, a whole ton of stuff, right? So all of the tongue rolling, I mean, yeah, I wouldn't even try and get that. All of those kind of simple things that you were taught in genetics in high school or even maybe in Gen Bio 1, they're largely all wrong. Like Widow's Peak, not single gene trait. So there's a whole, most things that you can actually observe in a human and pretty much everything else is polygenic. Multiple genes contribute to the phenotype that you observe, right? And that's simply because we are complex organisms. If you're looking at yeast, I mean, maybe you'd have a bunch of single gene traits, right? Single cell eukaryote, right? But we're not. We're multicellular organisms, so we're pretty complex ones too. So almost everything is controlled by many different genes. So clearly, Mendelian inheritance is not going to fly, right? It's not going to work. Well, I mean, it's not going to explain what we're actually interested in, is what I'm getting at. Okay, so classic is Snapdragon flowers. I've come to actually hate Snapdragons over the years, to be honest. Nothing against them personally, but it's just irritating. It's one of the very few examples of incomplete dominance that I can find. And it's just super annoying. So anyway, so essentially, when we talk about dominance, really we're talking about function. So there's two things actually. It's interactions between alleles, but if you go, a step or two further, really what you're talking about is function. Does that protein function? Does that protein exist anymore as a result of that mutation? And so typically when we talk about dominance, we're talking about that gene functions normally, or that gene functions normally and produces a protein that functions normally. And in terms of normal, it works, right? If it's an enzyme, it does its enzyme things. If it's a structural protein, it holds stuff up. It is expressed where it should be and when it should be and in the quantities it should be, right? All of which are very important to protein function and a whole bunch of other jazz, right? It's a complex subject. But essentially we're talking about normal when we talk about dominance. Doesn't always apply, right? You can have things that are dominant that don't work. It depends on the context. We're not going to talk about dominant negative mutations because it just kind of confuses me actually. So if it confuses me, it will probably confuse you as well. So typically when we talk about a recessive allele, oh God, what's my dog doing? We're talking about a version of the gene that doesn't function properly, right? Talk about dominant allele, we're talking about the version that functions normally. Often that is called wild type. And every time I have to use this in a paper, I have to look up whether or not there's a hyphen there. So wild type allele basically equals normal function, i.e. what you would find in the wild. And obviously that's a silly term to use in many ways, right? It's kind of, we do because it's habit. But if you think about it, out in the wild, there's a huge amount of genetic diversity, right? So which is the correct one? Which is the normal one? But either way, when we talk about wild type, we mean this is the one that functions properly, right? And so really it's about function, like loss of function or reduction of function means those things work less well. Gain of function actually is pretty funky. Those tend to be dominant because they're working when they shouldn't. But they could also be negative as well. Recessive. It's really funky. Actually, one of my main research is on the gain of function mutation. Actually, is what I do almost all of my research with, which is kind of fun. All right. Boop-a-doop-a-doo. Also, this depends on scale, right? So, and dominance isn't always complete. Now, the only examples I've ever found of this, and trust me, I've spent a lot of time looking for other examples of incomplete dominance that aren't snapdragons, right? This is like a personal crusade, I guess one could say. right? And I haven't found very many. Actually, in leopard geckos, because people do crazy breeding with leopard geckos. It's just eye-opening, because my daughter was really into leopard geckos for a while. She had one for a bit. There's actually, I think, an example of incomplete dominance there, but the genetics are so imprecise because a lot of this is amateur breeding that I never actually truly feel comfortable teaching about it as an example. So I'm not 100% sure about what's going on. So typically, though, incomplete dominance, where essentially one dominant allele is not enough, right? All the examples I've ever seen, of which there are not many, are due to pigmentation. So essentially, the deal with snapdragons and eggplants, I think they're called, aubergines. Essentially, the recessive phenotype is white. And so it's white because that's no pigment, right? That's an absence of pigment. And so if you've ever tried to paint a white room, like particularly red, actually, red is a really difficult paint to use. But anyway, another color. No, Cynthia, that's actually heterochromia. That's very different. I'll get to that in a second. You have to put multiple layers of paint on, right? Because otherwise there's patches of white that shine through. That's essentially what's going on here. So if you have one functioning allele, you produce some pigment, but there's not enough pigment in order to mask fully the recessive phenotype. So even though the recessive allele isn't doing anything, its kind of phenotype is still partially expressed because it isn't fully masked by one dominant allele. You need two. And so there's a couple of classic, I don't know, symptoms, outcomes of incomplete dominance. One, you have an intermediate phenotype in the F1. Heterozygotes, essentially. Right, and that's what you can see in this diagram on the screen. And two, if you cross those heterozygotes together as if you would in a monohybrid cross, right, you get, how would I put this? In the F2, the genotype and, my older dog is trying to escape the little pest. The genotype and phenotype ratios are the same. Equals a 1 to 2 to 1 ratio, which is what you can see down here at the bottom. So in a classic case of dominance, Mendelian dominance in a monohybrid cross, you get a 3 to 1 ratio for the phenotype and a 1 to 2 to 1 ratio for the genotype. Crap, I've got to go let the dog out. Give me just a second. That's not the case for incomplete dominance. Sorry about that. They can go terrorize each other outside now. Okay. And I haven't forgotten you, Cynthia. Still getting to that. And this is actually, that was a really good question, Cynthia, because it ties into codominance as well, which we'll get to in a second. And eggplants. Yeah, it's the other example, right? Again, a matter of pigmentation and the case where with a single functioning allele, you get an intermediate phenotype. Now, it doesn't really matter how close that intermediate phenotype is to one or the other. It's simply that it exists equals incomplete dominance. Because if it was complete dominance, the F1s would look exactly like the dominant parent. That's what I've been teaching you all this time, right? That's not the case with incomplete dominance. So getting back to Cynthia's question, that's a really cool question. So essentially, I'm not 100% sure as to the genetic basis for heterochromia, which is when you have two different eye colors. Sometimes you can actually have two different iris colors actually in the same eye, which is really wild, right? And actually you see this kind of thing in plants a lot. Obviously they don't have eyes, but if you think about variegated plants, right? Those plants that have two different colors in their petals, usually it's white and another color. It's like white and red roses and some other flowers, I guess. I'm not a botanist. Have that. Essentially what that is different lineages of the cells. So, you start off with one cell, divides into two, each of those divide into two, and so on and so forth. If a gene is turned off by mutation, or in female, right, humans, mammals, by X chromosome inactivation, all of the cells that descend from that original cell will have a different phenotype from all the cells which descend from the other cell that does not have that change. And so I know for sure in plants that's due to a mobile genetic element called a transposon, which basically jumps into a pigmentation gene and inactivates it, right? But that doesn't happen for all cells. It just happens at some point in the lineage so that you get those different patches. That's also not the same as spots on cats and stuff. So that's something completely different. So my guess, if I was to make a guess, and this is pretty much a wide ass guess, to be honest, is that it's due to a gene involved in iris pigmentation being on the X chromosome. And what happened very early on in development is in that individual, and I've actually only ever seen this in females. I haven't seen it very often, so I don't have a big sample size. So I don't know if it is sex linked, but one guess would be that it's due to X chromosome inactivation, right? Because you have two X chromosomes, Cynthia. I only have one, right? So to get the same amount of gene product from my X chromosome and your X chromosomes, you have to shut down one of yours in every single cell, right? And which one gets shut down is random. So my guess is that there's something involved in eye pigmentation, which is on the X chromosome. And so in heterochromic individuals, at some point, one X chromosome is shut down in one lineage, which leads to one eyeball or iris cells. And the other one is shut down in the lineage that leads to the other one. That would be my guess. I don't actually know for sure. But yeah, that's something very different. Actually, here's a good word. That's called mosaicism. Oh, it's definitely due to a mutation, but how that mutation affects the function and why it affects it in one eye versus the other, I don't actually know. Yeah, it is definitely due to a mutation, and also it's complicated by the fact that eye color is a polygenic trait, right? So and also it's a matter of chance as well, right? So this is actually one of the crazy things. I shouldn't talk too much about this because we've got to move on, but in development, it's all about lineages, right? So all of you every little bit of you, other than the bacteria in your gut and on your skin and stuff, has descended from one single cell, right? All of your cells, even though those cells are very, very different, like your bone cells versus your skin cells and so on and so forth, right, are very different. And so even though it's a little bit less precise in mammals than it is in worms, for example, all of your brain cells descended from one part of that tissue in a developing embryo, right? All of your gut and your lungs and kidneys and stuff. I actually haven't taught Dev Bio for a while. I'm a little bit rusty, but they all develop from a different group of cells, right? So essentially at various points, you have these divergences, right? Where a cell will divide and its descendants will develop into the gut, whereas the descendants will develop into the, I don't know, mesentery, let's say, right? And so if there's a change at one of those kind of branch points, so that one is different from another, and those actually develop into two different, sets of organs or whatever, you'll see a different phenotype in one versus the other. If that change occurs prior to that, both of those lineages will have that change. So you just have to think about stuff in a different way when you talk about development. It's kind of like a family tree, but for cells. Anyway, so the next thing to talk about is something called codominance, right? And I'm not going to use the examples in here because I kind of get tired of talking about peas, to be honest. And peas are not super, 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 super interesting. But essentially, codominance is where both alleles are expressed equally, right? So incomplete dominance is where there's not enough of one product to mask the other, right? So you're essentially not getting the full phenotype, the full dominant phenotype, you're getting a partial, right? Codominance is actually trickier to get, and it's very much more likely to be a molecular trait rather than something you can see with your eyes, because you can express both alleles at the same time. And so my favorite example, and I love talking about this because it's just super neat, is get out my teeny tiny whiteboard. As you can see, we have fall decorations up. Shortly to be Halloween decorations, and then shortly after that will probably be Christmas decorations. Like, I don't recognize my own home when I walk into it sometimes. Anyway, so codominance, no. That sucks. See if we've got another one. I'm going to do everything in purple. Well, I guess I will be doing everything in purple. Crap. My one black pen is gone. Okay. In blood type. I love talking about this because it's just super fun. And actually, I can't remember if I've ever been a recipient of blood transfusion. No, I don't think I have. Nearly did. Tore my spleen once when I was an undergraduate, which was not a very good idea. Yeah. My mum and dad got a call in the middle of the night saying, "Your son's in hospital, we think he's broken his neck." And they went, "Ah!" and all freaked out. And I wasn't actually that bad. Anyway, bit of a tangent there. So the cool thing about blood types is essentially you have cat fluff stuck to my fingers. You have your phenotypes over here, and I'm B positive, for example. So if you ever find me, crumpled in a heap and I need a blood transfusion, you can tell the doctors that, or the paramedics. Most people know what they are, and I know mine because I used to give blood when I lived in the UK. So you can have A, B, or O. are the 4 different phenotypes, essentially, that you can have. Right. This, and I've got to write really tiny because it's a tiny, tiny board. This is called the universal donor. So you can basically plug an O to O blood type person into anybody. You just stick them straight in and they'll be fine. We'll get to Y in a little bit. These AB, however, I don't know what you'd call these, universal acceptor, if you can even read that. Those are individuals that can receive blood from anybody. So that's the blood type to be. Does anybody know if they're AB? Just out of curiosity amongst the class. Does anybody know what the blood type is? Yeah, it's a useful bit of information. You should go give blood. It's a good thing to do. Save someone's life. And I don't know, I think, ooh, Cynthia, you're O. All right, everybody be friends with Cynthia. She could be very useful in a pinch. Anyway, and then there's also A and B. There's also rhesus factor. So I'm B positive. Cynthia's O positive. I don't actually fully understand that, to be honest. It's something to some maternal deal. And it's quite complex as well. I did try reading up on it once and I got terribly confused, so I stopped. So in terms of alleles, you can essentially go allele, genotype, phenotype, right? You have A, B, and O. And O is the recessive allele. In terms of genotypes, you can have a whole bunch, right? So AA, homozygous dominant for A, AO, equals A. Same for B. AB is another heterozygote, but now you have both dominant alleles together. And the AB phenotype is very different from the A or the B phenotype. If you give AB blood to an A or B individual, you'll kill them. That's why it's good to know your blood type, right? Obviously, when they go to give you blood, they'll check first. They're not just going to like try one randomly and see how they do. And O is the homozygous recessive phenotype. So essentially, you have 3 different heterozygotes possible. And they all have different phenotypes. I'll let you kind of cogitate on that for a little bit, and then I'll wipe it off and show you why that works the way it works. Also, so I can drink my coffee before it gets cold. It's a myth that my black pen doesn't work. Ah, dang it. Oh, quit it. This, there we go. Camera is such an old piece of crap. Actually, I don't even need to use it anymore. I've got a nice camera on my phone. Questions? Oh, under AB, it's universal acceptor. Sorry, Tiny Rising. Maricela, yeah, so the only way that you could be, well, hang on. If both your parents are O, the only thing you could ever be is O. Right, but if you're O or type O, you could be type O if your parents are any combination of these three. Hello, Jess. Oh yeah, thank you, Mika. Universal acceptor. Right, so if you have two, an A and a B individual, or two A's or two B's that are both heterozygous, then 1/4 of their offspring will be type O. If they're both O, then you can only ever have O, essentially. So it's actually kind of fun because you can, actually, unfortunately, my mum's dead, but I'll have to ask my dad to see what blood type she was. Don't know if my dad knows what blood type his is, but you can actually start kind of working back through your own family tree with this knowledge and kind of figure out, you know, what genotype your parents were and their parents and so on and so forth. So if you're kind of into like family tree type puzzles, it's a fun one to do. So the reason why, and this is actually just a fun thing to talk about, like as a biological process, right? A and B are antigens, right? They are, yeah, I don't actually, are they glycoproteins? Whatever they are, they're surface molecules that's on the surface of a red blood cell, right? So if you have type A, for example, you express the A antigen and have B antibodies. So in terms of blood type, you have the antibodies for whatever antigens you don't have. So if you're A, you have A antigens and B antibodies. So these are antibodies that you raise, you know, yourself. I don't know why actually, but you do. And so that basically means you can accept A and O only. Because if you were given B type B blood, then your B antibodies would stick to that blood and they would clog it up and you'd die from like basically a congestive failure. Your blood would clot throughout your body, which is not a good thing. And vice versa, right? So if you have B, I don't know what they actually look like. These are just kind of handy symbols to use. So antibodies are often shown to AB. You can accept B and O. Now, if you have AB, I'm just going to have enough space here. Doesn't that look pretty? You have no antibodies. So you're expressing both phenotypes on your red blood cells equally. So it's not that A dominates over B or B dominates over A, or that you have a partial phenotype. You have both phenotypes, right? And so these, because they have no antibodies, can accept blood from anybody. However, type AB individuals can only give blood to people that are also AB or O. Because if they give blood to people that are A or B and you have these antigens on the blood cells, they'll be recognized by either the B or the A antibodies in those individuals. Adriana, that's a really good question. I got to say, I really don't know, and I've tried to understand this before and I've not succeeded. The rhesus factor has a big deal with during pregnancy. I don't actually understand why. Sorry. I know it's yeah, so Adriana, I know it's something to do with another antibody that is produced, right, which can react between the mothers and the embryo due to antibodies crossing the placental blood barrier. But I don't actually know really why or what it is. It's kind of confusing. Cynthia, you're welcome. You're correct. If you're AB, you have the AB antigens and no antibodies. That bit there. And so if you have no antibodies, you can accept blood from anybody because you will not react to the antigens on their blood cells because you don't have antibodies to them. But you can't give blood to everybody like type O. And so if you're O, you have A and B antibodies. Oh, and another key point as well is that when you give blood, the red blood cells are stripped out of the plasma. So all the antibodies and other stuff that's soluble in your plasma stay behind. That's why you can give blood cells and not worry about the antibodies. And so if you are type O, and this is probably the downside for Cynthia, so you can be a universal donor, but you better hope there's another type O person kind of around if you get in trouble, because you can only accept blood from O individuals. So actually, if you are type O, blood donor banks will love you, right? Because they're fairly rare, and that's pretty much it in terms of what you can have if you're a type O individual. You know, it's crazy. My mum actually gave over her life over 100 pints of blood to the British red blood thing. Oh, so you've already experienced that, I guess. Yeah, I imagine even more so in the military as well. But yeah, type O individuals are exceptionally valuable individuals. Obviously, everybody's valuable, right? We treasure everybody. But in terms of trauma and ERs, type O people are literally lifesavers. I actually never really minded giving blood. I always went there for the cookies and the hot cup of chocolate afterwards. You didn't get paid in the UK, you just got fed. So I'd be quite happy there, just kind of lying with my blood draining out of me and eating cookies and drinking a cup of hot chocolate and chatting with the nurses, some of whom were rather nice. I was a teenager then. So anyway, that's co-dominance. Sadly, I can't actually give blood in the US because I grew up during the mad cow epidemic which actually happened before most of you were born, which is a little bit bizarre, in the UK. And so there's no test for Creutzfeldt-Jakob disease, which is what caused the cows to go mad. And so they won't take my blood here. Because I may have the ability to turn mad, but I just haven't turned mad yet, essentially, is the issue. I'm pretty sure I would have if I was going to, but anyway. Okay, so cracking on. Now, this one is what I would call a doozy, which I know that most people don't actually know what that means. It's actually a very complex and interesting phenomenon to talk about. So epistasis basically means that the phenotype of from one genotype, one gene, can mask or affect the phenotype expressed by another gene or locus. So essentially, you can have different genotypes and express the same phenotype because one of those genes controls or masks what you see from the other gene. That's really what epistasis is about. And there are lots of different forms of epistasis. And really what we're talking about, generally speaking, are series of events, right? So something relying on something else for you to see the outcome. And the classic symptom of epistasis is if you do a standard dihybrid cross, because there's always at least two genes, you'll not get the 9:3 to 3:1 ratio. That's a very nice thumb there, Jess. Very large. Maybe you want to see a doctor about that. Anyway, so get back there. Of one gene will mask the genotype of another. And you can, and basically what that means is you won't, sorry, typing issues. I don't know what's going on. I think my trackpad is being awkward. You won't get the standard 9:3 to 3:1 ratio. You'll get something else, right? And so here's one example of that, right? So this is negative epistasis. And so essentially negative epistasis is two genes in the same pathway. And you need both to see, or both functioning. To see whatever that pathway produces, right? So for example, in this case, we've got two enzymes which together turn some precursor into a pigment. And it's a multi-step pathway. And to be honest, most pathways, either whether enzymatic or signaling or metabolic or whatever, most of those, if not all of them, have more than one step. So you typically start with something, and you turn that into something else, and then something else, and something else. and then you end up with the product at the end that you want. So if that product is a visible one, then you have to have both genes functioning to see that visible outcome. Right, so for example, here, actually, remember I was saying I'll never give you an exam question where in a dihybrid we have parents that's homozygous dominant for one and homozygous recessive for the other? Well, here's an example of that. So you can actually have two parents that are, pardon me, white, cross them together and you'll have a purplish offspring, right? Because it's heterozygous for both. So it has, so here the standard law of dominance applies, right? So we have this isn't intermediate or co-dominance where we get this kind of like funky issues of dominance. Dominance applies normally here. So that if you have a functioning allele, a dominant allele for both genes, both enzyme C and P, you'll get purple. Right. However, if you're homozygous recessive for either of these, that phenotype masks the genotype you have for the other gene. So if you, let's kind of blow this up a touch. For example, right? This individual here, or this one, actually any of these white ones, they all, for one of the genes, are homozygous recessive. Now for the other gene, like enzyme C, right? For example, this one here on the left that I'm circling, which is what, like the second square, is homozygous dominant for one of the enzymes. So it's like fully functioning, great, you know, 2 copies of the dominant allele, doesn't matter. You will not see the purple phenotype because for the other gene, that genotype is being masked. And it doesn't matter in this case which way round that is. So elsewhere, for example, you could have a dominant allele for the second enzyme. But if the other enzyme is homozygous recessive, or the other gene rather, you're not going to see that genotype get expressed. Right, so this is called negative epistasis. Right, and so where you have this situation, you'll have, instead of a 9:3 to 3:1 ratio, you'll have 9, right? So those are all individuals that have at least one dominant allele for both genes. And then all the rest, the 3 to 3:1, those all get clumped together, and that's just 7 recessive individuals. So that's a very big departure from the 9:3 to 3:1 ratio. That's not the same as linkage, which we'll get to maybe today, if not next Tuesday. That's about alleles traveling together because those genes are close to each other on the same chromosome. There you'll get something sort of looking a bit like a 9:3 to 3:1 ratio, but it won't be. Here you'll get a very definite 9:7 ratio. And then you can also get crumbs. There's lots of different types of epistasis. You can get dominant epistasis. Actually, my favorite example is white cats. So white cats are white because they have a dominant negative mutation in something that results in the cells that produce pigment in cat fur. And so you can have a cat that is genetically a black cat because it has a dominant allele for the black pigment gene, but it will be white because it's homozygous for the dominant white gene. So the genotype you have for white masks whatever genotype you have for pigment color. You'll never see it if you're white. The only way you can see color in cats is if you're, sorry, if you have essentially that white gene is not dominating stuff. And that's also why you have patchy cats. So it's somewhat white in some places, and then you see the pigment in other places. That again is another example of mosaicism, and there's a lot of that in cats. Anyway, so that's epistasis. Epistasis is actually, it's super interesting, and it's really a great way of working out gene order. So if you have a dominant mutation, and this is essentially most of my work in, and this isn't an enzyme pathway, but it's a signaling pathway. So if you have a dominant mutation in this one, which results in a different phenotype, in my case, worms have too many vulvas, which is kind of unfortunate. It's called multivolvar phenotype, or MUV. Like little cows, they've got these kind of bobbles sticking out of them. But if you then get rid of gene function of genes downstream of that, you don't see that gain of function phenotype. And so if you get rid of something downstream and it gets rid of the phenotype that you were seeing, that tells you that that gene is in the same pathway and downstream of the gene that you're interested in. So you can do this, and you can basically, and this is what people have done for this particular pathway, you can discover and order all the different genes involved in that process simply by doing these kinds of experiments. Oh, so Bridget, I'm not an expert in Harry Potter, although I've watched all the films and read all the books. Interesting question though. So maybe whatever her name was, can't remember her author, didn't have a thorough grounding in genetics before she wrote that. The two magic folks having a non-magic kid is easy. If we assume that, actually, I guess this depends on your perspective, right? Is being magical the dominant phenotype or the recessive phenotype? That's the question, because depending on which way around it is, depends on which of those is going to work or not. So if we're assuming that because the vast majority of people in the UK are not magical, even though we're special, then maybe you'd assume that being magical is recessive, right? It's a recessive phenotype. So in that case, you can have two non-magical parents produce a magical offspring, because those non-magical parents could be heterozygous. Well, they both have to be heterozygous, right? And so they could have a homozygous recessive offspring as a result. Now, if, again, we're assuming that being magical is recessive, you cannot have two magical parents have a non-magical offspring. Right, because if they're magical parents, then they both must be homozygous recessive, so you can't have anything but a homozygous recessive child. And that, well, actually, it could go either way. But that's also really what the whole, shoot, what was his name? The guy with the long white hair. No, not Dumbledore. The nasty guy with the kid that was in the school. Blonde, not white, blonde hair. Draco Malfoy, that's it. Was it Lucius Malfoy? I think. There you go. I got my wife just correcting me from upstairs. How silly is that? She's the expert. Yeah, she's saying yes. So anyway, that's what essentially lies behind the whole pure blood deal. Right, so essentially that's the same. It's about breeding, right? And so they're arguing that essentially being magical is a genetic trait which you inherit, right? And so you want to be pure blood because that means that you're maintaining that lineage. You don't want any, scruffy non-magical alleles coming in. That's a bit of a simplification. And obviously there's probably some flaws that I could dig out there if I wanted to when it comes to genetics. But so no, actually, who asked that? Bridget. That's really that's actually a classical Mendelian inheritance, not non-Mendelian. You'd have to do an awful lot of breeding of wizards and witches and non-magical people to figure out anything more than that, which even in the Harry Potter world would be unethical, one presumes. Okay, so what's next? You're welcome. It's always fun to think about these things from a different context, right? So this actually, this thing of complementation, this is actually in many ways more of a technique than it is a phenomena or phenomenon that one would explore. So essentially what we're talking about here is how do we figure out if mutations that we've discovered, or if we get a bunch of individuals with the same phenotype, actually, like say we're doing a mutant screen in worms or mice or plants or whatever, how do we figure out if all those phenotypes are the result of mutations in the same gene? Because again, think about the context that we were looking at in epistasis. These processes that we're looking at are most likely the result of multiple steps, pathways. So if you do a mutagenesis screen, you could end up with actually probably what you're hoping to end up with is mutations in lots of different genes that are all involved in the same process. But how do you figure that out, right? You could have just ended up by chance with a whole bunch of mutations in one gene and nothing in any other. So to test that, what you do is you do what's called a complementation test or assay. And essentially what you're asking is, can I get the normal phenotype back or not, right? And so if your mutations are all in the same gene, right? So you've got allele A and allele B, same gene, and you cross those two together, now you will get what's called a compound heterozygote, which is a fancy way of saying it doesn't work, right? So actually, this is a fun term. Don't use it very much. And it's not something I tend to teach as a concept because it gets kind of funky and complex. But this is actually an issue in things like Tay-Sachs disease, which is very prevalent in Jewish communities. And so one reason why they do a lot of genetic testing is you can actually have individuals that have different recessive alleles, but if those two recessive alleles come together, you will still have the disease phenotype. Because even though they're different alleles, neither of them functions. And that's called compound heterozygosity, which is fun words. So if you still have the mutant phenotype, when you cross these two different alleles together, that tells you that they are in the same gene, right? And obviously you can have multiple alleles in the same gene, not restricted to just one, right? You can have lots of different changes that result in a non-functioning gene. However, if they're in different genes, right? So we have a mutant allele in one gene, and because we've done a lot of work to make sure we just have that one allele present, right? You do a lot of things called outcrossing, right? So you maintain that mutant allele, but get rid of all the other junk that you might have accumulated in the mutagenesis screen, right? For every other gene in that organism, it's going to be normal, bar the mutant allele in that gene of interest. Now, if that's in a different gene, then the opposite will also be true. For the B allele, it's going to be normal for the A gene, right? So when you cross those two together, you'll have a heterozygote for each, right? So you'll have a dominant allele for each gene, and as a result, you'll have normal function. So that tells you that A, those alleles are not in the same gene, and also those two different genes are most likely involved in the same process. So then with that information, then you'd go back and you'd start doing epistasis experiments, right? We've got two different genes. What order are they in this process, right? And then you start trying to figure that out. So this is more of a technique than it is something that you would be interested in and of itself, right? This is a means to answer questions in genetics rather than the questions themselves. And so it's funny, the Dr. Page who developed all these PowerPoints, and I've been kind of changing them, over the years to suit my own personal interests. But he actually did a lot of developmental biology in axolotls. So he's a real reptile expert and stuff. So anyway, that's what these slides are here for. But I think I understand it well enough to kind of look at it. So essentially, axolotls are super weird organisms, as you probably know. And they're essentially called pedomorphs. And so typically reptiles will go through a juvenile to adult molt, where like tadpoles to frogs, for example, right? And axolotls don't ever change into the adult. So even though these are reproductive adults, they still display the juvenile phenotypes like external gills, for example, and goofy expression, I think, as well as another part of that. So when you cross these different types of axolotls, it's called a tiger salamander, right? Essentially, what you're asking the question is, are the mutations that cause this phenotype, are they in the same gene or not? And so when you cross them together, you end up with the wild type phenotype, which is a normal non-juvenile adult, right? It doesn't display the juvenile phenotype in the adult form. And so that tells you that whatever's causing the pedomorphic phenotype here is different from the one that causes it in this one. Because when you cross them together, you have dominant versions of both genes, or dominant alleles of both, even though they're heterozygotes. And so you exhibit the normal adult phenotype, which is kind of a fun thing to think about. So really it's about how do you ask those questions in genetics? And complementation is one way in which you do that. They're all essentially, it's like a toolkit, right? You have all of these different kind of ways of asking these questions, which are super, super neat. So kind of ripping through, we still have a little bit, but not too much. Maybe we'll finish it today, maybe we won't. So these are really interesting, right? So particularly when it comes to disease, right? And so essentially you can have variable expressivity, right? And that's really basically saying the degree to which you express that phenotype varies, right? So you can have two individuals with the same genotype, right? And so they should have the same phenotype, but in some cases or some traits, you'll have an individual with a very mild version of that disease and an individual with a very strong version of that disease. This isn't, don't get confused with like infectious diseases, like COVID, for example. Some people get really bad COVID, some people get like barely a sniffle, right? That's down to a lot of complex factors, including your immune response and other predisposing factors. This is more talking about genetic diseases, right? So ones which are based on having non-functioning versions of particular genes. Actually, in COVID, I think it's because some people have immune systems that work too well, right? Actually causes a lot of damage. So in a lot of diseases, you can actually do yourself more harm than the disease does by trying to get rid of the disease. And that's, I think, in one way in which COVID tends to kill people. And so that's this different phenotype for the same genotype, or different degree of phenotype, is called variable expressivity. Now that's different from what's something called variable penetrance or incomplete penetrance. That's the case of you have the same genotype, but you don't have the disease. Another individual has the same genotype and does have the disease. So not everybody with that genotype has that disease. And so actually a really good example of that are the BRCA1 and 2 genes that we talked about earlier in the cell cycle checkpoints. You know, if you're homozygous recessive for both, 70% of individuals will have or will develop breast cancer. 30% of individuals with the same genotype will not. So the penetrance of that is said to be incomplete, right? If you have that genotype, it does not mean you're guaranteed to have that disease. Probability is very high, but it's not 100%. So that would be an example of incomplete penetrance. And actually, for a lot of, you can kind of think about that in terms of lifestyle diseases too, right? So if you smoke like 60 cigarettes a day, there's a pretty good chance you'll get lung or mouth cancer or esophageal cancer or something nasty by the time you're like 60 or 70 years old. But it's not guaranteed, right? Again, that's probably not a great example, but it's a good way of getting a handle on it. We'll talk about polydactyl later with cats. Actually got a student cat to show you. So kind of ripping through these a little bit because these are fairly straightforward in some senses and extraordinarily complicated in others. And so basically the vast majority of traits that you will look at or that you can observe are complex or polygenic traits. And so there are multiple genetic factors at the very least, but also gene by environmental interactions. Classic one is height, right? So you can have height is determined by a whole bunch of different genes, right? Genes that affect, you know, kind of spacing along your anterior posterior axis as an embryo, bone development, muscle development, things like that. But also, you know, just from looking at individuals from, you know, drought-struck or famine-struck countries, that they'll also be shorter. You know, even though they may have the potential to be tall, they don't have the nutritional availability to develop to that potential, right? So most traits have that kind of multi-gene and gene by environment interaction. And just to kind of finish that off super quick. Oh, and another classic example of polygenic traits is you tend to get an even distribution of phenotypes. Height is a classic example of that because there are many, many different genes that contribute to that. So normal distribution often equals polygenic. Binomial is less likely so. But another thing as well is that, and if you've ever done any plant breeding, which I haven't, but I know people that do, you can actually select for different traits in different environments, right? And there are always trade-offs too. I know I'm going a little bit over, but I just want to finish this up. So for example, if you go out into, I don't know if we grow much wheat in Texas actually, but there's an awful lot of farms kind of around San Antonio. I think they just grow cows. But you can select for cows, right? Which do really, 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 really well. When there's a lot of water, there's a lot of feed, they're constantly doused with antiparasitics, and you get these mahoosive like cows that you get a lot of steaks from, right? But if you put those same cows into, I don't know, India or Africa, they'd be screwed, right? They'll most likely not survive, or they'll be very emaciated or whatever, right? So that's a gene bioenvironment interaction, and you've essentially selected for some traits at the expense of others, right? Whereas if you look at the cows that you get from like sub-Saharan Africa or India, they'll never grow as large, but they're much more robust and resistant to changes in environment, right? So in that case, selection has or whether human or natural, has selected for a more versatile collection of alleles with regards to, what matters to them. Anyway, that's it for me. I know that was a little bit of a rush at the end, so we could spend more time on fun stuff early on, like Harry Potter. I've got to get like Star Wars and stuff into here somewhere. That's kind of more my cup of tea. But anyway, next class we'll dive into linkage. So if you can read ahead, it's a real brain twister, lots of math involved. So thank you very much, everybody. That was a whole bunch of fun, and have a lovely weekend, and I'll see you next week.